All of the information that we know about biology, or that we think we know, has been confirmed by experimentation and the study of living things. So, what I want to do in this lecture is to answer the question of how are living things studied. And the caveat is that there are a lot of variations on how living things are studied. We're going to look at the scientific method, and we're going to look at it very carefully with specific steps. But really, the scientific method can be deviated from, and it often is. So while we'll go through kind of a careful examination of using the scientific method, know that there are many ways to interpret the steps and many ways to use this method. Scientific method begins with a question about organisms. It can be about behavior, adaptation, structure. It can be about different functions within the cells. But ultimately, these questions about organisms lead to testable statements called hypotheses. And hypotheses are tested by measuring the validity of predictions. Predictions are if-then statements. So often, scientific method studies will describe hypotheses as if-then statements. But really, if you have a hypothesis, it is a statement that doesn't usually include an if and then. Instead, the if-then statements are used for testing the hypothesis. So we'll look at examples of that. In 2001, there was a study published in Nature by the Finnigs, and the Finnigs were studying mimicry. Batesian mimicry was long established for probably a century, where the idea was that non-toxic organisms mimic the patterning or the coloration of toxic or lethal species to avoid predation. And they used a snake system in the Carolinas to carefully test whether or not Batesian mimicry was at work in populations of these snakes. So the observation, the long-known observation, was that some harmless animals mimic patterning and coloration of related poisonous animals. Their test organism was the scarlet king snake, which is a non-venomous snake that lives wherever you see just plain yellow. Here, this is the range, the habitat of the king snakes, the scarlet king snakes. And that range extends into a range that is also inhabited by the eastern coral snake. So dark orange is the range of the eastern coral snake, and notice we have this overlap of the two snakes. Hypotheses that were formed by the Finnigs could have been, and these weren't the exact hypotheses, the exact statements from the paper, but a hypothesis could be that king snake predation decreases in areas where coral snakes are present. Now, there is another way to state a hypothesis. Very rarely is it used in common descriptions of the scientific method, but the null hypothesis is a hypothesis that's a little bit easier to test and a little bit easier to confirm. In this case, the null hypothesis is king snake coloration has no effect on predation. Now, this is the opposite of what we think is happening, right? But we can easily test whether or not the king snake coloration has effect on predation. And we can easily confirm whether or not this null hypothesis is true. So our if-then statement to test the hypotheses, if king snakes are not patterned to avoid predation, then predation of king snakes will be constant, whether or not the habitat of the coral snakes overlaps. So the idea is that the predation rate of the scarlet king snake will be constant, whether we're talking about an area where there is not eastern coral snake overlap or where there is. The if-then statements anticipate the results of some kind of experimental or observational study. And that study will yield data which does or does not support the hypothesis. So the study that the Finnigs did involved creating artificial snakes. They created two artificial snakes. They created an artificial king snake which has the exact patterning of the scarlet king snake. And then they also used the compounds that they used to make the king snake that's fake, to make an artificial brown snake. And this brown snake does not have the coloration of the king snake, the artificial king snake. The artificial snakes and brown snakes were placed in areas where coral snake habitat overlaps. They were also placed in areas where the coral snake habitat does not overlap. And then the predation on these fake snakes, just the fake snakes, was determined by evidence of attack by small animals. So here is some evidence of an attack by predation. Now, their study had to include controls. These are important to account for unintended influences or some kinds of alternate explanations for the results. The data will not inform the accuracy of the hypothesis if it's the result of any kind of unintended influence or any kind of alternative explanation. Now, variables, there are a few types of variables. In an experiment, the independent variable is the one that's varied or manipulated by the researcher. So the conditions that the researcher puts on the experiment. We can also think of it as it's the variable that the experimenter assumes to have a direct effect on the next variable, which is the dependent variable. And that's the response that's measured. So the dependent variable depends on the independent variable. This is the one that the experimenter measures after making changes to the independent variable. Now, the controlled variables are variables that could change and have an indirect effect on the experiment, and they could have an indirect effect on what we're measuring. These are controlled and made consistent by the experimenter. Okay, so the question is, what are the independent and dependent variables in our described experiment? What is manipulated by the researcher and what is measured? In our case, what we know we're going to measure is the predation on the snakes. The independent variable is the presence or absence of coral snakes. So it's not whether or not our fake snakes are colored or not, because that didn't have anything to do with the hypothesis. The hypothesis had to do with presence or absence of coral snakes. And so what the experimenters controlled was the habitats that they put these fake snakes in, equal numbers of brown and striped artificial snakes in both areas where there is no coral snake and where there is coral snake. And the results. Here is the area where coral snakes are absent, so where just the king snakes are present. Here's where coral snakes are present, so where the habitats overlap. Here on the y-axis is the percent of total attacks on artificial snakes. And we're going to look at both the striped and the brown snakes. We have the independent variable on the x-axis of this graph, the dependent variable on the y-axis of the graph. The dependent variable is, in this case, measured in percent, and that's really nicely labeled here. And then these bars represent the percent predation on the two types of snakes. So where coral snakes are absent, we have 83% of the artificial striped king snakes attacked. 17% of the brown type are attacked. So probably these are much easier to see, of course, than the brown snakes. So 17% could be seen and attacked. Now let's look where the coral snakes are present. 84%, excuse me, I shouldn't say this. I think I said that this is of the snakes. This is 83% of the attacks. So the two of these columns are 100% of attacks. Now, of all the attacks, 84% of them were brown snakes, and only 16% were the artificial striped snakes. So when we were in an area where the predators knew about coral snakes, the percent of total attacks on artificial king snakes dropped significantly. where predators don't come into contact with coral snakes, these brightly colored snakes were much easier to attack and were attacked more often. So, predation is significantly reduced in regions where the coral snake habitat overlaps, and our null hypothesis that the coloring has nothing to do with predation is nullified. We have officially strongly confirmed that the coloration does potentially have something to do with predation. So what I want us to take away from this is the steps of the scientific method and also the difference between the different variables. Controlled variables in this type of experiment would have been things like placing the snakes in areas where ground cover was the same, so some weren't more hidden than others. Making sure that all of the studies were done maybe at the same time of year, and with the same numbers and types of predators in every place where the different artificial king snakes and brown snakes were placed. And I'm sure you can think of other things that had to be controlled as well. Percent that were laid out, percent of the different artificial snakes, the abundance of predators, all kinds of things we could think of for controlled variables. All right, so now what I want to do in our next module is to move into talking about how living things are built. We're going to start talking about structures.